Marcus here on urine formation. Blood enters the kidney through the renal artery where it eventually comes into contact with a nephron, which is where all of the magic happens. Step 1, glomerular filtration. So blood enters the glomerular capsule through the afferent arteriole, where it comes in contact with the filtration membrane. It's made up of 3 parts. The fenestrated capillary, fenestrations in the capillary themselves keep things like albumins and red blood cells in the capillary bed. That basement membrane, it's very negatively charged, repels anions and keeps them in the capillary bed, keeps them in the blood. And filtration slits, which further filter the blood and produce filtrate. So filtration slits are formed by podocytes. When one podocyte nudges up to another podocyte, you have this little gap, this little slit, and that's a filtration slit. So filtrate is formed and eventually goes into the proximal convoluted tubule. Now, how much filtrate is formed, glomerular filtration rate, is calculated by NFP, which is essentially blood pressure and the surface area in the glomerular capsule, the surface area of the filtration membrane. Here is a juxtamedullary nephron. Here's the glomerular capsule of the juxtamedullary nephron, and here's the distal convoluted tubule of the juxtamedullary nephron. Here we have the macula densa, which kind of sends how much what the GFR should be. Essentially, I'm not going to get into it. Only 5 minutes. Here we go. Step 2, reabsorption. We have the peritubular capillary surrounding the proximal convoluted tubule. Reabsorption primarily occurs in the proximal convoluted tubule. It happens through a few different mechanisms. We have passive diffusion, transports things like water, sodium, water, ions, salts, urea, uric acid, and bicarbonate down their concentration gradient. That's just the way that the direction that it wants to go, since we removed all that stuff from the blood earlier. Endocytosis, and that is more active type of transport. Any proteins that somehow manage to slip by the filtration membrane, amino acids, because they're larger, and then co-transport. This is done with an ion like sodium, will attach to a protein and kind of push things like glucose, amino acids, lactate, and phosphate along into the interstitial fluid and back into the blood. So here we have step 3 and 4. Step 3, the secretion mainly happens in vasa recta and the peritubular capillaries if you're a cortical nephron, the vasa recta if you're a juxtamedullary nephron. And secretion is the removal of any toxic waste like ammonium or creatine, uric acid, urea. These things are secreted in the proximal convoluted tubule, and ammonia is primarily secreted in the distal convoluted tubule. So water conservation happens in the loop of Henle, nephron loop. Henle named everything after himself. The nephron loop. So this mechanism happens when ADH activates albumins and kind of forces them to the surface of the nephron loop, allowing water to exit. Because this nephron loop is not permeable to water unless aquaporins attach themselves to the outside of the nephron loop. So ADH kind of keeps water in our system. If we didn't have ADH telling these aquaporins to go to the outside of the nephron loop, then water would stay in there and we'd pee it all out. So distal convoluted tubule. Then we go to the collecting duct, where urine is finally happening. More water conservation is happening as water exits the collecting duct by osmosis, and then we get our final product and pee it out into the toilet or elsewhere.